Dr. Ames, our main speaker for tonight. He's a professor emeritus of molecular biology and biochemistry at the University of California, Berkeley, and a senior scientist at Children's Hospital Oakland Research Institute. He was also a member of the National Academy of Sciences and was on their Commission of Life Science. He was on the board of directors of the National Cancer Institute, the National Cancer Advisory Board, from 1996 to 82. His awards include, this long on my arm, lots of awards. The General Motors Cancer Research Foundation Prize, the Tyler Environmental Prize, the Gold Medal Award of the American Institute of Chemists, the Glenn Foundation Award of the Gerontology Society of America, and the Honda Prize of the Honda Foundation in Japan. And it goes on and on. He's also received awards from the Linus Pauling Institute, the Prize for Health Research, and the American Society for Microbiology Lifetime Achievement Award in 2001. The Thomas Hunt Morgan Medal for the Genetic Society of America, and the American Society for Nutrition Rose Award in 2008. He has more than 540 peer-reviewed publications and is in the process of working on 3 right now, I believe, from our conversation at dinner. Dr. Ames invented an inexpensive assay to check for mutagenicity of compounds. Since then, he's focused on understanding the biochemistry of aging with a focus on mitochondria and how micronutrients may prevent disease, malnutrition, and obesity. Good, definitely have things going now. He was instrumental in studying mitochondrial health and mitochondria's role in longevity and disease prevention. Dr. Ames also developed the triage theory. A triage is from the French word to sort, and it's used on battlefields by doctors to prioritize treatment so that efforts can focus on those with the highest priority. Dr. Ames had observed the approximately 33% children coming into his clinic with obesity, and he has been working at the Children's Hospital in Oakland with the African American and Hispanic populations. The obese lacked essential micronutrients such as magnesium due to poor nutritional intake. So he will be talking about nutrition and aging and how we can improve our nutrition and prevent aging. Here you are, Dr. Ames.
I think I'm wired up, so I may not
Okay.
need Can you hear me all right?
Yes.
Yeah, we're going without this for a couple of minutes.
Okay.
And that will go to the next, I will go to previous, and that's an ordinary pointer.
Okay.
But we have to get both screens up.
So I used to be chair of biochemistry at Berkeley and then retired in my 70s because they wanted to redo our building and cut everybody's lab in half and move us somewhere on campus, and I moved to Children's Hospital of Oakland Research Institute. And I'm going to be 85 in December, but I still have a big lab, and we're doing the best work of my career. Anyway, my mitochondria decaying ever more rapidly, but I finally figured out what they're decaying into. Hypochondria. Okay, first, could we see?
You may have to look at that eventually. I can't get the screen up.
Oh, okay.
Hang on. Here's PowerPoint.
That, it was up before.
Yeah, I can't get both screens on, but you may be able to present from that one. Okay.
There, there it is. Okay.
You'll have to use that screen with the whole this one.
Okay. So, this is your metabolism. So, you have 20,000 or so enzymes converting one small molecule to another, and cycles and all that. But what do you need? What do you need to run your metabolism? Well, you need fuel, and you get that from burning fat or carbohydrate in the mitochondria, and you make ATP, which is the high energy fuel of the body. And then you need about 40 or so micronutrients. And these are compounds that you need in your metabolism, but the body can't make, and you have to get them from your diet. And that's what I'm going to discuss. What are these, and are we getting enough? And the answer is no. Okay. So these are roughly, there are a few micronutrients people argue about, and I won't get into all those arguments, but roughly you need about 15 minerals and 15 vitamins. And then there are the essential amino acids, histidine and tryptophan, etc. And then there are 2 fatty acids you need, the omega-3s and the omega-6s. And we should be getting close to 1-to-1 or 2-to-1. And now we're getting 17-to-1 because we're not getting enough omega-3s. And where do you get your omega-3s from? Deep sea fish. So 30% of your brain is DHA, which is a long chain omega-3 fatty acid. And if you eat your tuna and your salmon, you get plenty of it. You can make it somewhat inefficiently from the flaxseed oil has an omega-3 and a few other vegetable oils, but really it's better fish or a fish oil pill or a krill pill if you want to get it from that. And Americans really aren't getting enough. And then, so what I'm going to talk to you about is mainly the 30 or so vitamins and minerals. And you've heard about all these things, but I think people aren't thinking straight about them. So the committees, the government sets up committees to come up with numbers, how much should we be getting of this? But everything's based on short-term disease. And what I'm going to tell you is we should be basing it on long-term health. So, and are we getting enough? It's clear from this that we're not. Menstruating women are losing a lot of iron, so 16% of the menstruating women are below the EAR, which is the official deficiency point. So EAR is something called estimated average requirement, and that's the number it's some distribution in the population, and if you're below the EAR, they say you're deficient. And they set the RDA at 2 standard deviations above the EAR. So really, there are 2 linked numbers, the EAR and the RDA, but for deficiency, you should look at the EAR. And magnesium is a complete disaster because 56% of the US population is below the EAR, and nobody seems to care because they don't have a pathology. But I'll tell you what the pathology is going to be. It's going to age you faster. So where do you get your magnesium? It's in the center of the chlorophyll molecule. So nature's colored it green. Magnesium isn't green, but it's in the center of anything green. And you eat a big plate of kale or spinach, you're getting a good dose of magnesium. Now, you get a little from nuts. Nuts are healthy food, but there are not too many foods that are high in magnesium. And magnesium is like calcium. Minerals are tricky because calcium and magnesium are one above the another on the periodic table. And minerals all look similar. So calcium has 2 plus charges and magnesium has 2 positive charges. And it's hard for nature to completely differentiate them. So if you take a calcium pill and put a big dose of calcium in your body and you're not getting your magnesium, you can trash some of the magnesium enzymes and vice versa. So really, nature cares about the ratio, and you should be taking a calcium-magnesium pill. Anyway, you can overdose yourself on minerals very easily because too much of one mineral might interfere with another. Too much iron interferes with zinc or copper. Potassium and sodium are very similar. Again, they're above each other on the periodic table. And people say, we're all getting too much sodium. But one of the reasons we get too much sodium is we're not getting enough potassium. And again, the body cares about the ratio. So minerals you have to be careful about. Mae West said, too much of a good thing is wonderful, but I think she was thinking about sex, not micronutrients. So you don't want to overdo it. And zinc's in 2,000 enzymes in the body, and they zinc fingers turn on a particular sequence in DNA. It's in copper zinc superoxide dismutase, and it's in P53, which is mutated in half of human tumors. And yet we have 12% of the population below the EAR for zinc. And vitamin B6 moves amino groups around and decarboxylates and transaminates. It's involved with the synthesis of all the neurotransmitters. And again, half the elderly women are too low. And folate, does anyone know folia, what the Latin word folia means?
Leaf.
Leaf. Foliage is the same root. And my mentor at Caltech, when I got my PhD in the early 50s, first isolated folic acid from 4 tons of spinach. In those days, you needed a lot of material to characterize something. Anyway, so you eat your greens, you're getting folic acid. You eat your greens, you're getting magnesium. And I'll also talk about vitamin K, which you get from your greens. So, but a good varied diet gives you all of these things. And then vitamin E, we're all pretty low. And vitamin C, about 1/3 of us are too low. And then they hadn't set EARs at the time I did this slide, and we're very low in vitamin K, which comes from greens, vitamin D, which you need from sunshine, calcium, potassium, and omega-3. So should you be taking a multivitamin mineral? And I think everybody should as insurance. But on the other hand, it's not a complete thing. You should eat a good varied diet, and fish oil is not too expensive. You can take a fish oil pill if you're not eating fish to get the omega-3s. And then one a day or a multivitamin mineral doesn't have enough potassium because it would make the pill too big. So you need about this much potassium and a fair amount of calcium and magnesium. So they put a token amount of calcium and magnesium in the pill, but essentially no potassium. So eat a lot of veggies and you're getting your potassium. So you could eat a banana in the morning, you get a 0.5 gram of potassium. Okay. So, let's see. I'd like to tell you about an experiment that got me involved in vitamins and minerals. Some years ago, I had a guy come to my lab on sabbatical, Jim McGregor, who was a scientist at the USDA lab. He was a cytogeneticist. He was looking at chromosome breaks and things like that. And someone came up with a neat test for looking at mutagens that broke your chromosomes. That's the most dangerous lesion from radiation is when you break chromosomes. And so it turns out you all know that red blood cells don't have any DNA. to make a red blood cell, the precursor cell, you extrude the nucleus, and then you make a red blood cell. But someone found if you stain red blood cells for DNA, in a mouse, 1 in 2,000 lights up. There's a little piece of DNA in there, a micronucleus it was called. But it's due to if you break a chromosome and there's a piece of chromosome floating around, when you extrude the nucleus, it gets left behind. And that's a measure of double-strand breaks, the really bad lesion. You irradiate the mice to get more of them. So McGregor it's a very easy assay to do. You can just put a fluorescent dye that binds to DNA and the cell lights up. And you just count them, 1 in 2,000 lights up with mice. And he was studying what interferes with this. Does caffeine was supposed to inhibit some repair enzyme? Does it inhibit double-strand break repair? And he was doing dose response curves with radiation. And one day, all his control mice were full of chromosome breaks. And he yelled at his technician, "Did you irradiate them by mistake?" And she said, "Absolutely not." So McGregor's a very good scientist, and he tracked this down. He examined every item of the food, and he called up the company that made the vitamin mix for the mice. And they said, "Oh, Dr. McGregor, we're going to call you." Turns out somebody left folic acid out of the vitamin mix. And so he tried folic acid, and if you lower the folic acid, you get chromosome breaks just like radiation. So he thought that was pretty interesting, and he studied that a little bit, and just then he came to my lab on sabbatical. And so I was blown away by this experiment. I thought, gee, that's really neat. And then McGregor wanted to look in people. But this whole test doesn't work in people because the human spleen, which is down here somewhere, I'm not quite sure where a spleen is, but anyway, the function of the spleen is all your blood goes through the spleen, and any cell that's a little stiff gets weeded out by the spleen because nature doesn't want some clogging up a capillary and getting a thrombosis or something. And the human spleen works very well, but all these inbred mice don't have any splenic function, which people knew, but it only turned out later it was important for this test. So McGregor looked at people who'd been irradiated, he didn't see anything. But there are a lot of people who've lost a spleen in an accident. So he went to Kaiser, and they have 6 million people in their database, and he said, "How many people do you have without spleens?" And they said, "Oh, 50 or so." And so and they live perfectly well. So he went and got permission to get blood from 20 of these guys. And lo and behold, he could see these broken chromosomes, the basal level. And that's this experiment. And this is an absolutely gorgeous experiment. It was published in 1988, and it should have changed nutrition, and it's just buried in the literature. I think nobody's looked at it except me. But it changed my life, and so I've been working on vitamins and minerals ever since, because I was interested in DNA damage, and if a lack of folic acid could give you DNA damage, that could be really important. It's just like irradiating the people. So anyway, what he did is he looked at these 20 people, and he got a baseline down here. This measures these chromosome breaks, micronuclei. But one guy had this huge level of chromosome breaks. He said, "What's wrong with this guy?" And he couldn't, the fellow seemed pretty normal. He wasn't eating a great diet, but it didn't seem horrible. So he followed him for a year and tried to figure out what was wrong with this fellow. And he bounced up and down, but he was always very high compared to the baseline. And then when he discovered this folic acid business in mice, he said, "I wonder if he's low in folic acid." And he was low, but not wildly low. 10% of the population was at that level, and 50% of the poor was at that level. But he gave him some folic acid. And this is his wham. In 2 days, it was down to here. He gave him folic acid for about 10 days. And the reticulocytes are the newly synthesized red blood cells. It's about 1% of the red blood cells. And you can fish them out with magnetic beads, and they're newly synthesized, and those are the ones that came right down to baseline. And red blood cells take a long time to turn over, and those slowly came down to the red blood cells. And then he ran out of this folic acid, and both of them started shooting up. And he said, gee, it's unethical to do any more experiments on this guy now that I know he's breaking chromosomes because he's not getting his folic acid. So he said, are you going to eat a better diet, or do you want a pill? And the guy said, give me a pill. So he gave him a pill and said, you better take this. As long as you're eating this bad diet, take it forever. And the guy went away. But he followed him for a bit, and here's his micronuclei level came down just like everybody else's. But he measured the serum folate, he measured the red blood cell folate, which is much higher than the serum. Red blood cells are not only delivering oxygen to the tissue, they're the FedEx trucks of the body. They're just filled with vitamins and minerals and essential amino acids. And when they dump the oxygen and pick up CO2, they also dump all this other stuff. So they're very high in everything. So it's easier to measure the red blood cells. Anyway, so he thought this was pretty important, and he looked up the numbers, and 10% of the US population was at the level where you're breaking chromosomes, and 50% of the poor. The poor eat a god-awful diet. And and he had just come to my lab on sabbatical, and I got all excited by this experiment. Hey, why are we worried about a part per billion of pesticide? I never thought that was very credible anyway. We should be worrying about what we're leaving out of our diet. And he wasn't a biochemist, and I helped him work out the mechanism. We published a paper on that. And one of the postdocs in my lab compared folic acid to radiation, folic acid deficiency to radiation. And Fenwick in Australia did an experiment about the same time. I'll show you Fenwick's. We published 2 papers in different journals. But Fenwick, I'll show you his slide. So Fenwick looked at the folic X-rays is the white, and the more X-rays, the more chromosome breaks. And then the less folic acid, you cut it in half, you're already significantly higher, and then just goes up. So people worry about incredibly low levels of radiation for Fukushima, and they're not worried about eating the greens. And all this was published back in 1988. And again, people in nutrition really didn't get it. But so, and it helps to work out the mechanism, and we think we've really worked that out, though it's not 100% pinned down. So the 2 main pools of folic acid, the methylene the form in the body is not folic acid, that's an oxidized form. It's tetrahydrofolate. When Herschel Mitchell, my mentor at Caltech, first isolated folic acid, they worked up all this and they oxidized it. Folic acid is easily oxidized, and they got oxidized folic acid, and that's easy to synthesize, so that's what's in vitamin pills. And the body can reduce it, not terribly well. So I think probably they should have methyl tetrahydrofolate in the vitamin pills. But that's quite easily oxidized, so you have to pack it in the right way. Anyway, that's, but methylene tetrahydrofolate was discovered at Berkeley some years ago, and that methylates uracil to thymine. Uracil's in RNA, thymine's in DNA, and the methyl group doesn't change the base pairing, but it's a tag telling your cell that that's DNA and that's RNA. And then you have an enzyme that converts this pool to this pool, and methyl tetrahydrofolate plus vitamin B12 convert homocysteine to methionine synthetase. And this is one of the standard amino acids. This is just an intermediate getting there. But methionine goes to something called SAM, S-adenosylmethionine, and that's the main methylating agent in the cell, one carbon units. It makes choline for your neurotransmitters and your lipids, and it makes all sorts of other things. So it methylates DNA, but every time it does that, it regenerates homocysteine. So you really are cycling this all the time, and you need these two vitamins to complete the cycle. Otherwise, homocysteine accumulates. And there have been many papers showing homocysteine is associated with cognitive dysfunction if you get high in homocysteine. And also it's associated with heart disease. I won't go into all the details on that, but that was the story. And the mutation, about 40% of Northern Europeans have a slightly different form of this enzyme, methylene tetrahydrofolate reductase, that converts this to this. And that mutation increases this pool at the expense of this pool. Now, if you don't have enough of this, you get uracil in your DNA, and that causes nicks and breaks, and it's not, there are enzymes all the time clipping it out. And cancer, several types of cancer have been associated with low folate. And if you don't have enough of these vitamins, homocysteine accumulates, and it gives you heart disease and cognitive trouble. So why would Northern Europeans do this? Okay, well, I think nature cares more about your sperm, and then heart disease and cancer come late in life, and there's not a strong selection for them. But sperm, nature really cares about. And so 4 or 5 types of cancer have been associated, and we published a paper waving our arms and doing a few experiments showing that you get more damaged sperm when you were low in folate. So at least it's a plausible explanation why this mutation, most polymorphisms, that's alternate forms of genes, are probably due to some kind of selection pressure. And I think a lot of them are going to be nutrition, but we'll see. Anyway, so we were reasonably satisfied we had a plausible mechanism, and so why folate deficiency causes cancer. But then that raises the question, well, what about deficiency of all these vitamins and minerals? How many are giving you cancer, whatever? We're not eating a good diet to get them all. And so the mechanism is just like radiation is dangerous because you get clusters of electrons from radiation. And when you have a cluster, you can damage 2 bases, one on one strand and one on the other. And when you have 2 nearby lesions, the radiation biologists think that's the most dangerous part of radiation, because repair enzymes are always cruising along the DNA, clipping out lesion damaged bases that don't look like the normal bases, but the other strand is holding it in. But if you have 2 nearby ones, then you get a double strand break, and that's really hard to repair. And so the radiation biologists have a consensus that's the most dangerous part of radiation, and you get that when you don't get your folic acid, or B12, in fact. So then the next micronutrient I got involved with was Fernando Viteri, who's a Guatemalan who is a professor of nutrition at Berkeley. And he knocked on my door one day and said, iron, I'm an expert on iron, and the World Health Organization, iron's dirt cheap, it's just rusty nails. The world, and there are 2 billion women and children in the world who don't have enough iron, and the World Health Organization is going around giving iron to everybody. I said, that's a good thing. He said, no, it isn't a good thing because they're giving them too much. That too much iron is bad for you. And he said, would you help me do the biochemistry, and we'll look at the whole range of iron in mice. And it was known that too much iron, too little iron damages mitochondria, and we were experts on mitochondria. So one of my postdocs, Patrick Walter, and one of Terry's graduate students, Mitch Knudsen, did the whole range of iron. And so this is in mice, and this is too little iron, and what we're measuring is 6 different things about mitochondria. Mitochondria efficiency, mitochondria getting oxidized, mitochondria oxidative damage, and other things. And the red line is the sum of it all. They're all, so too little iron, and that's 2 billion women and children in the world, is bad for you. It fouls up your mitochondria. And too much iron is bad for you, and you should be in here somewhere. So metals are really tricky. And if you tell people selenium's good for them, which it is at certain concentrations, 5% of the population is going to go out and poison themselves by taking too much selenium. So you just have to be careful, because vitamins have a complicated structure, and enzymes can tell one from another very easily. But minerals are difficult, and too much iron is probably causing a zinc deficiency or a copper deficiency. We're not sure, but anyway. And then the next, I had a postdoc, Emily Ho, who came to my lab, and she was a zinc expert. So she took human cells in culture and made them a little zinc deficient. And this is measuring oxidation. Fluorescein is a wildly fluorescent dye. And somebody made a reduced form of fluorescein that's not fluorescent. When you oxidize it, it becomes very fluorescent. It goes into cells. And you just can measure this easily in cells. If cells are full of oxidants, you get all this fluorescent cells. And so zinc deficiency does that. So the mitochondria are starting to pour out oxidants. And then there's a COMET assay, which measures DNA damage. I won't go into the details, but basically you put nuclei on a microscope slide and put a charge across the slide. And if the DNA has nicks and breaks, it pulls out, and then when you stain the DNA, most cells look like this, but if there's nicks and breaks in the DNA, it pulls out and it makes a comet-like thing, so it's called a COMET assay. It's a very easy assay to do, and people use that for measuring chromosome breaks and things like that. Well, zinc deficiency does it. And so all these oxidants are mutagens, and they'll break your chromosomes. And one of the other postdocs showed when you made human cells in culture biotin deficient, they fill up with oxidants. And we think, we've done several of these, vitamins and minerals, and we think the oxidants are coming out of the mitochondria. They're doing the nasty work. So what mitochondria do is they burn the fat and carbohydrate, and in that burning, in the Krebs cycle, a citric acid cycle, you're bringing the fat or these sugars down to these Krebs cycle acids, and that allows you to pump. And these 4 complexes do the passing of the electrons. You're pulling electrons from the fuel and adding them to oxygen with the electron acceptor, and you get water. So, and in the process, you release energy and you pump these protons across the inner mitochondrial membrane. And what that does is put a charge across the membrane. So mitochondria are like little batteries, except you're recharging them all the time by burning your fuel. And that potential across the membrane allows complex 5 here to convert ADP to ATP, so it makes this high energy molecule. You're making a kilo of that in your body every day, of ATP, and it powers your muscle, your brain, and all your uphill biochemistry. So lots of Nobel Prizes were awarded to all the people who figured all this out. If the system isn't perfect, then you release a 1 electron here and there. So what complex 4 does is add 4 electrons to oxygen to get water, and then your home's safe. But if you leak an electron here or there, you get superoxide or hydrogen peroxide, and they're all oxidizing agents. So mitochondria are always putting out 1 or 2% oxidants. And as you get older, it gets worse and worse. And you'll find out soon enough all your systems are degrading a little bit. As I say, my mitochondria seem to be turning into hypochondria, but everybody's different. Anyway. This shows magnesium deficiency, and that's over half the country, is cross-links mitochondrial DNA to protein. So you get radicals and cross-link it. David Kilway in my lab showed that. And so I wondered, why is nature doing that? Why is nature giving you DNA damage in all these cases? And it didn't seem to make sense. So I put in the 30 vitamins and minerals into Google, and next to it I put mutation, cancer, chromosome breaks, and out popped all this literature. So calcium deficiency, Fenwick had shown gives chromosome breaks, and Lipkin showed you get colon cancer, and folate deficiency, McGregor and us, and Fenwick all showed chromosome breaks in mice and humans, and Willett had shown cancer, and vitamin D deficiency has been associated with many types of cancer, magnesium deficiency and zinc deficiency, potassium, B12, selenium, omega-3s, niacin, choline. So I said, that's odd. Why is nature doing this? And one day it hit me. I had this epiphany. That's just what nature wants. Because through all of evolution, nature's been running out of vitamins and minerals because the minerals aren't spread evenly through the earth. There's some areas high in selenium and people get poisoned because they're getting too much selenium, and there are areas low in selenium, people get poisoned because they're not getting enough selenium. Selenium's involved in biochemistry, and et cetera, et cetera. Iron, red soils have a lot of iron. Soils that aren't red don't have so much iron. And then plants have their own vitamins and minerals, but people migrate and they don't eat the same diet. So, so I was thinking nature must have come to grips with this. And what I postulated just from this kind of circumstantial evidence, that there's what nature does is when it gets a little low on any vitamin or mineral, it does a strategic rationing. What it does is the proteins that are need that substance for working and are essential for survival get it first. And if it's DNA repair or helping you protect against calcification of the arteries or something that's long-term, some insidious damage, then those proteins suffer. They are disabled. So that means we're thinking about vitamins as acute short-term effects, scurvy, beriberi. But in fact, vitamins involved on both short-term things and long-term things. Humans live 80 years or more. Lots of them are needed to keep you with a long life. Anyway, I call this triage, this kind of rationing, and wrote a paper, a theoretical paper about it, and got all sorts of vitamins are associated with DNA damage or other long-term things, and threw it out there. But I can't say the nutrition community has adopted it. People are very conservative, and you have to fight for years. I did the same thing when we were trying to convince people that DNA damage had something to do with cancer. And, oh no, it's viruses, or no, it's this or that. And finally, when everybody comes around, they say, oh yes, it was obvious all along. And then you know you've won the battle. But anyway, this was the paper back in 2006. Most of the world's population has an inadequate intake of one or more micronutrients. We posit that as a result of shortages, natural selection developed a metabolic rebalancing response to shortage. the rebalancing favors short-term survival, and you pay a price in long-term health. And this insidious damage results in the disease we associate with aging. Cancer is insidious DNA damage that accumulates, and eventually it makes some key mutation and you get cancer. And I'll talk about a few others of these. So cognitive decay and all these things. So the thesis is that the price you pay for not eating a good diet is it ages you faster in some way. And I'll give you a few illustrations. Now, Joyce McCann used to be a postdoc with me 40 years ago, and she came back to the Bay Area looking for a job, and she's really smart. And she's very analytical, and I'm more intuitive, so I hired her on the spot. And she wrote 4 big reviews about the developing brain in the fetus, and vitamin D and iron and other things. So you really need a complete nutrition for the baby. You need a complete nutrition for your brain. Anyway, she wrote 4 beautiful reviews on that. And one day she came into my office and said, "Bruce, I'm skeptical of your triage theory, and I think there's a way to test it. What we want to do is analyze vitamin and minerals that are in the literature that have been well studied but aren't too complicated. You go after zinc, the 2,000 enzymes, you'd spend the rest of your life in the library." So I said, "Joyce, go to it." And so she picked vitamin K as the first one, and her analysis is really beautiful because there are all these medical implications. So I'll just run you through that. So these are the ones we thought weren't too complicated, but it takes about 9 months to do one of these reviews, and reading all the papers and writing and getting it all put together. And so she did one on vitamin K and then one on selenium, and then we figured, well, we're proving it to our satisfaction. We published the paper, and the rest are going to be the same because it makes biological sense, and let somebody else do it. So anyway, so she did one on vitamin K and one on selenium. These were the ones that weren't too complicated. Vitamin K is in 16 different proteins, and selenium is in 25 or so proteins. So they're something you can handle with 9 months in the library. Anyway, so what does vitamin K do? So vitamin K is something called phyloquinone, and where do you find it? It's used in photosynthesis in plants. Again, nature's colored it green. Anything green has vitamin K in it. And Americans don't eat a lot of greens, so lots of us are short of vitamin K. And this phyloquinone is very lipid soluble, so when you eat some greens, it goes into your lipoproteins and is delivered to the liver. And so how do you tell which proteins are essential and which proteins are more long-term? And so what Joyce did is she started off with a crude look at it. She said, well, vitamin K was discovered in Denmark by a guy named Dam, and K stands for coagulation. You spell it with a K in Danish or German. And so you needed something in greens, otherwise your blood wouldn't clot. And they isolated this vitamin K. And what it is, it's a coenzyme for a protein that takes an already made protein that has a glutamic acid in it, which has one acid group on it, and put another acid group on it so it can bind calcium. All these 16 proteins are calcium binding proteins, and they all have this funny amino acid called GLA in it. So it's a particular sequence, and the enzyme knows to do that, but you can't do it without vitamin K. So a lot of these proteins had been studied for years, but nobody knew that there was a GLA in it till somebody discovered that. Okay. So then it's vitamin K dependent. So we don't really, if you knock out any of these in mice, and Kopecky won a Nobel Prize for figuring out how to knock out any gene in a mouse, and you don't make the protein. And so then somebody wrote a paper, what happens when you knock out this gene or that gene? Well, if you knock out any of these coagulation proteins that are involved in blood clotting, And that's clearly essential. You cut yourself once every week or two, and you'd bleed to death if you didn't have coagulation. And when you're making a mouse, you're making 20,000 miles of capillaries or something like that. And if you didn't have, if there's any little imperfection, the mouse would die. And that's exactly what happens. They're embryonic lethals. The mice never survive if you knock out that gene. So they're essential for survival. And that makes sense. And then you look at the proteins that are not essential. Those are the interesting ones, because those are the ones that get trashed. And so osteocalcin, you can guess from the name, it's binding calcium. These are all calcium binding proteins, and it's in the bone. And you knock it out in mice, and they're born, and they look okay, but you stress them a bit, the bones break. So you can make a bone, but you can't make a strong bone without vitamin K. So bones aren't just calcium and vitamin D. You need this osteocalcin too. And these are the ones that play second fiddle, because since they're long-term, if there's a limiting amount of vitamin K, it goes to the blood clotting protein. And then, and it's known that there are more bone fractures in people who are short of vitamin K. And 30 million prescriptions are written every year for warfarin. It's also called Coumadin. Anybody here take warfarin or Coumadin? Yeah. So that's about, in any crowd, there's one or two people. If you get thromboses, that can kill you. So those are little blood clots in your blood. And so the doctors try and thin your blood, which is what they do is interfere with blood clotting and interfere with this vitamin K system. But what they don't quite realize is you're trashing all these proteins. So people who get warfarin, Coumadin, get many more bone fractures. And then this Gas6 is involved with diabetes. It's a tyrosine kinase receptor. Anyway, it's an interesting protein. We haven't done too much on that. Matrix GLA protein is very interesting. Because you knock that out, and the animals are born. The mice look perfectly okay, and then they all drop dead. And what do they drop dead of? Calcification of the arteries. So it turns out calcification of the arteries starts with a calcium phosphate crystal. Calcium is all over the body, and phosphate's all over the body. But calcium phosphate crystallizes if you look at it. It's a very insoluble crystal, and it's called hydroxyapatite. The geologists call it. And that's the start of an atherosclerotic plaque because it interferes with the blood flow, and then the cholesterol coats it, and pretty soon you have an atherosclerotic plaque. Well, the docs don't know this, so all these people getting warfarin and Coumadin get a much higher rate of having calcification of the arteries. But we didn't do any of the work. We just were theoreticians coming in and reading all these papers, or maybe Joyce McCann. And there's a very good group in Holland doing all this work, and a good group in Boston. And they're trying to get to the bottom of all this. So, but these are the proteins that are causing trouble. And matrix GLA protein has another interesting aspect in the way the body does this priority. It sends the vitamin K to the liver, and all the coagulation proteins are done in the liver. But these other proteins are not done in the liver, they're done in other tissues. But the liver gets first crack at everything. And then the vitamin K is converted to another form of K. It's called MK, menaquinone, rather than phylloquinone. And that, if you feed menaquinone, it gets to all the tissues equally. And the Japanese have a health food that's full of menaquinone. It's a menaquinone derivative, MK7. And this is natto. Has anybody ever eaten natto? Yeah. Oh, a well-traveled group. So I'll get to that in a minute. So osteocalcin, so you look at the function, bone structure, and mouse knockout, fragile bones, and human mutants, You can look at human mutant. You can look at the human gene, which Joyce did, and ask, are there any mutations in there that have some pathology? And in fact, you get brittle bones if you have mutations in the human gene, so that fits. And then anticoagulate therapy gives you worse bones, and modest vitamin K deficiency affects your bones. And similarly, matrix raw protein, the negative regulator of vascular calcification, mouse knockout arterial calcification. Human mutants is a rare genetic mutant in the human gene that's associated with people dying of calcification of the arteries in early age. So that, again, that fits. So, and arterial calcification is higher if you're getting warfarin and vitamin K deficiency is. That gives you that. This is what natto looks like. It's a Bacillus subtilis fermented soybean. And the Japanese, you get a plate of it for breakfast. Looks a little yucky. There are these long white strings. And it smells a little yucky, and it tastes a little yucky. So Westerners just don't like it. It's like eating Limburger cheese for the first time. Not too many people like it, but you can get used to it after a while. And it's considered a health food, and all the evidence in Japan on the epidemiology is that people get fewer bone fractures, they get less cancer, one of these is to do with cancer, and it all fits. Anyway, for what it's worth. Let me go. So TGF-beta was a TGF-beta inducible, was a protein they studied for many years, and then somebody found out there was a GLA in it. So it binds calcium, and it needs vitamin K. And that protein, the animals are born, they look okay, and then they drop dead of cancer. And it turns out that protein is in the matrix communicating with integrin on the surface, which communicates with the microtubules, and your mitosis doesn't work quite as well. So you may have a mitosis, you can distribute the chromosome, but you lose a chromosome here or there, and you get an extra one there. And that's associated with cancer, and all these animals die of cancer. So what's beautiful about this is you can understand the mechanism, you know how to deal with it. Now you can do epidemiology. Epidemiology is such a difficult science because they get the wrong answer half the time, because it's just some kind of an association. So you always should take epidemiology with a grain of salt. So Joyce did selenium. I won't go through all of that, but again, it's involved with these long-term things. Here's cancer, DNA damage, heart disease, reduced resistance to infection, muscle weakness, poor cognitive function. So again, some of the genes are essential for survival, and some are these long-term. So, and in these two big reviews Joyce did, about half the proteins were short-term survival and half were long-term. I call them longevity proteins. So basically their main function is to keep you with a long life, but you don't see an immediate effect, which then suggested to me that maybe there's a whole new set of vitamins that are involved with proteins that are only longevity. And we're looking for those, and we found a few, and I can briefly tell you about that. But anyway, I'm just writing a big review about that now. So, and selenium works in a completely different way than vitamin K. How it prioritizes, some proteins get it first. Selenium's in a transRNA and inserted into protein, and there's a modified base on the transRNA that determines whether it reads certain genes or not, and it's a clever way of doing it. So each system has developed some way for doing this triage priority. So what about the immune system? Well, you trash your immune system when you're short of vitamins and minerals. And which part of the immune system? Well, what goes wrong with aging in the immune system? These 6 things go go wrong with your immune system when you're old. Has anybody looked at this for shortage of vitamins and minerals? And the answer is yes. And we did all this in Google. Google's wonderful for all this kind of stuff. And somebody looked at vitamin A and someone looked at zinc. And this was a paper of mine that I completely had forgotten about. I had a French-Canadian postdoc who was looking at folate deficiency, and she was looking at part of the immune system, and the CD4-CD8 ratio of T cells changed on folate deficiency. I didn't understand it, she didn't understand it, but we wrote it up and published in the Journal of Immunology. And since it didn't fit with anything else in my mind, I promptly forgot completely about this paper until it showed up on the Google search. And then tryptophan, zinc, vitamin C, vitamin E, zinc, vitamin B6, vitamin E, tryptophan, zinc, vitamin B6, vitamin E, zinc. So people have looked at particular vitamins or shortages, but it makes sense because all your adaptive immunity is using a lot of energy and vitamins and all that. And if you don't remember the virus that hit you 4 years ago, it's better to trash that because you don't need it for survival. So this is all more long-term stuff. And that's what nature does. Oh, and vitamin D is really a hormone. Vitamin D, you can get it if you eat a lot of fish and deep sea seafood, but most people are getting it from sunshine. And it's 7-dehydrocholesterol is converted with a little ultraviolet light from sun into the precursor to this vitamin D hormone. And when they sequenced the human genome, there were 900 genes with that telltale sequence that vitamin D receptor binds to turn on the gene or turn off the gene. So it's a very important hormone, and you better get yourself normalized for vitamin D. And most of us are at our computers or watching television or playing video games. Kids aren't out playing in the streets anymore as much. And you can look at the vitamin D level, it's just been going down over the years. But the brain is absolutely filled with vitamin D receptor, and all sorts of enzymes in the brain require vitamin D. So there are 900 genes it's turning on. And what determines whether you get your UV ultraviolet light? Well, everybody's slathering themselves with sunscreen. The dermatologists say, stay out of the sun. But in fact, you need the sunlight to make vitamin D. And particularly if in Chicago, you're in trouble because it's dark. And if you have a melanin in your skin, you're also in trouble because you need melanin is a defense against too much ultraviolet light, which burns your skin. So an Irishman wouldn't last very long in Somalia or in Africa. And racially completely different people, Southern Indians are just as dark as Africans, New Guinea people. So dark skin is a positive selection in the tropics. Now, you can get tanned even if you have a light skin, and that's an adaptive response. But when you're in Africa or in Southern India, you're in sun all year round, and you develop a permanent tan, the dark skin. But just the way an Irishman in Australia is in deep trouble, and what they need is a hat and sunscreen, they have the highest skin cancer rate in the world because all these Brits and Irish, there's no sunshine in Ireland to speak of, and they all go off to Australia and get cancer, skin cancer. So now they're aware of it and they lather themselves with sunscreen and things. But if you're an African American in Chicago, you're in deep trouble. You need a lot of sunshine or take a vitamin D pill. And vitamin D pills are cheap, and you can, now the RDA, first of all, they fortified food, but they fortified the wrong food. They put it in milk, And 75% of African Americans are lactose intolerant. And they put way too little in anyway. Now the RDA keeps on going up. And so I think everybody should take, who doesn't get enough sun should take a 4,000 or 5,000 RDA pill. They set the upper limit at 4,000, which most of the experts in the field thought was kind of low for an upper limit, and I take 5,000 myself. But you need a better dose, and particularly if you're not out in the sun all the time. And if you have a dark skin, you have to be really careful about it. This shows DNA damage. It's a slide of Fenwick, and this is men, non-smoking healthy males and non-smoking healthy females. Women live longer than men, so they have less chromosome breaks. But look at that variability. Why is that? I bet it's diet. So if you don't get your vitamins and minerals, you're in trouble. And lots of genes are going to lead to cancer, not only vitamin K and folic acid. We have so many protective systems. Magnesium's in every DNA repair enzyme, and half the country is short of magnesium. So, and the worst diet is in the obese.
The worst diet is what?
In the obese. Well, let me summarize TRIA. So it's a mechanism of how moderate VM deficiency increases the risk of disease of aging and suggests a prevention strategy. Indicates about half the proteins analyzed are longevity proteins, which suggests that biomarker assays for setting EARs should be on one of the vulnerable proteins. And we're just working with people who are, you can already, there's a company that, run by a scientist friend of mine, which can take a finger prick of blood and measure 1,500 different proteins in that finger prick of human blood. And in a year or two, he's going to have all 3,500 proteins that express themselves in human blood. And I have another entrepreneur friend, scientist friend, who's put a machine in 2,000 different hospitals in China. The Chinese have subsidized him to do it, that measures a dozen or so proteins that are important for human health. And again, you put your finger in the machine, get a finger prick of blood, and you can do this. So we're working with both of them to try and get nutrition into all this. So I see the future as you're gonna, doctors don't know anything about nutrition, and they've completely abdicated. And so, and they're expensive. So the future is gonna be a cheap machine where you put your finger in, they'll charge you $10 or whatever, and they'll analyze the proteins in your blood, and you'll say, you're low in magnesium or you're low in zinc, and then you have to tune yourself up. Eat your kale or take a pill. Anyway, that's gonna come. I won't see it, I'm pretty old, but you guys will see it. And so the conventional wisdom is that obesity is too many calories in and too little exercise out. This was a cover of an Economist. But I think that's a simplification. This is the exercise out. So it's more, obesity is more than that. The blacks and the Hispanics are leading the charge and are getting really fat. And every possible disease that's been looked at has been linked to obesity, a disease of aging. More cancer, more brain dysfunction, more poor immune systems. Practically everyone that's been looked at it's accelerated in the obese. And there are 4 or 5 papers saying when they look at the diet of the obese, it's the worst diet in the country if you define worse as ratio of calories to essential vitamins and minerals. So they're starving. And what they're starving for is calcium and magnesium and zinc and all these vitamins. And I think nature makes you hungry when you're starving, so that it overrules satiety signals, and there's some evidence for calcium for that, so that these people are hungry all the time, their diet doesn't have the thing, and they just get fat. And it's a complete disaster. This is the American diet. This is top source of nutrition for 20 to 30-year-old, percent total energy. Sugary soft drinks, number one. A Coke and a Pepsi are a disaster. 40 grams of sugar, they punch holes in your gut, you get bacteria leaking through, you get inflammation. It's not good for you. And so it's not only what you're leaving out, it's not having a peach has the same fructose and glucose, and a peach is perfectly okay because it has fiber and it has other things that counteract this process. Anyway, people are working all those things out, but there are no greens on this list. Sugary soft drinks, pizza, beer, hamburgers, white bread, cake, donut, French fries, potato chips, rice, cheese or cheese fries. 40% of calories. So you want to give up processed, as you guys all know, you give up processed foods. Eat fruit for dessert, not cake. Give up the white stuff. Minimize the potatoes and the white rice and the white bread, and eat whole grain bread, and more fish and less meat, less red meat. And your mom told you all this. So the moms who didn't tell their kids to eat the veggies were selected out by evolution. So I stole this from some British website and changed it a bit. So being obese isn't good for you. Shape up, run the marathon, or maybe that's too extreme, but get some exercise. And I have an Italian wife, so I eat a wonderful Mediterranean diet, but she's nagged me all the time to get more exercise. So about 10 years ago, we were thinking about how do you get vitamins into the poor. You can give them a lot of pills, but the poor are well known to not take their meds and sort of drift and don't do what they're supposed to. And getting to take pills has been a problem. And the NCI has been telling them for 30 years to eat more fruits and vegetables with zero success. So anyway, we decided we'd make a bar. The local USDA was making a fruit-based bar. And we said, could you add a few vitamins to it? And it slowly developed, and we finally got a bar that tastes good after some years of struggle. And now it has all the vitamins. It's like a super vitamin pill. It has soluble fiber, insoluble fiber, all the polyphenols in plants, all the vitamins and minerals in fish oil. Anyway, we figured out how to get all these in and make it taste good, and a little chocolate and a little blueberry. And it does wonderful things to people. We call it the Corey Bar. I work at a place called Corey, Children's Hospital of Oakland Research Institute. And we've done 15 human clinical trials, and we keep on modifying the bar based on the clinical trial. So this is HDL cholesterol. The bottom of the arrow is where the person started, and the top of the arrow is 2 weeks later. One, you can see this is more HDL is good for you. So this is risk of disease, and it's pretty, not everybody, but it's pretty consistent. And we've done this over and over again. Now, all this was done on a shoestring. It's really, clinical trials are expensive, and we're always limited in money because you can't get funded for innovative science anymore. They're only funding 8% of grants, and if you're innovative, it's death. And I'm at an age where I don't want to do a 2% advance, so I'm just struggling for money all the time. Anyway, it works. And we've been able to improve antioxidant glutathione. We've been able to improve insulin resistance, and we've been able to improve LDL and HOMO-IR, the insulin resistance. And so we have a Mediterranean diet in a bar. And the nice thing is, we want to find out what's in there that's raising HDL. we can take out this ingredient or that ingredient. You can't do that very easily on a Mediterranean diet. And say it's a combination of 3 substances, and they're synergistic. So diet is hard to untangle, but we can deconstruct our bar. So I'm very excited about this. We're just looking for funding now. And I'll finish up now. And so every year, life expectancy gets longer. This is women, this is men. Women live a little longer than men if they're both getting good diets, and it's up to about 80 now. And people say, oh, the Europeans are living longer than the Americans. That was one of the selling points for Obamacare. But it's that living long isn't a good measure, and if you correct for auto accidents and you correct for murders, the US is living just as long, if not longer than Europe, and we do better on most medical tests that you look at. So it was a specious argument. Anyway, you're 57 years old, I'd like to get that down a bit. And this is the group. Jung Suh is of Korean background, but he was brought up in Thailand because his father was a businessman. And he's discovered a new vitamin. He does metabolic assays, and he's a member of it. We do a lot of things as a group. And Joyce McCann makes the trains run on time, and she's in charge of the Corridor project. And Swapna is an Indian postdoc who's our immunologist, and David looks at all the minerals at once, and Harold is a physician who delivers, he's a neonatologist, he delivers premature babies, but he works in the lab a couple of days a week. And Sandy is a forensic biologist, so she pioneered all the methods in mitochondrial DNA for identifying people, so she's getting calls from the FBI all the time. She developed tests and things. So you can lick an envelope and they can identify that it's you. Just a cell or two is enough. And then Mark is my Buddha. He's a Japanese American with a big round face and a big round belly, looks like Buddha, and he's our gut expert. And Michelle was a pediatric cardiologist who led our clinical trials and now is at the Children's National in Washington. And Ash Lal is an Indian hematologist who's our main MD now in the group. And so, thank you.
Thank you.
Discovered that play a role in longevity.
Yep. So, well, half the proteins were with half the proteins were with long-term in the selenium and vitamin K. So we started thinking there must be vitamins that have never been discovered among proteins that need them as cofactors or whatever, or special antioxidants that are only long-term. And there are lots of them. I'm writing a review about these. For example, carotenoids are the beta-carotenes, the orange color in carrots. Trees turn orange in the fall because the chlorophyll's gone. And every plant is out in strong light, and what carotenoids do is if you have light and a dye, that energy gets trapped in the dye and passed on to oxygen, and you can get a very energetic form of oxygen called singlet oxygen, which has a lot of extra energy, and it's a really powerful oxygen in the cell. And the defense against that is carotenoids are orange because they have all these conjugated double bonds, and the extra energy of the singlet oxygen gets dissipated as heat. So every plant has to have carotenoids. And we eat them in our diet, and we know we need beta-carotene. It's the precursor for vitamin A. But the macula of the eye has two strange carotenoids, lutein and zeaxanthin. And all the eye people know that they prevent macular degeneration, which is one of the main things doing in your eyes. Why are they there? Well, they're what they trap is blue light, and that's the most dangerous part of light for making singlet oxygen. And you can make singlet oxygen in the dark, and there's a pump that brings it to the macula. So it's a vitamin. We can't make it, we have to eat it, and there's a pump that brings it in, and it's just a longevity vitamin. So I'm writing a review saying, hey, we're talking about vitamins in the wrong way. We should include lifespan, and then they're much harder to find, but this fits the bill. Let's say I had a question. There's.
Here's my.
Oh.
You mentioned Kudaman and the thickening of the blood that is caused by vitamin K.
Yeah.
Is there a natural way to get rid of a thick blood?
Okay. So, well, vitamin K is natural. It's the compound warfarin is based on a compound in clover, I think, that made cows bleed to death, and that people in Wisconsin Alumni Research Foundation, that's why the name warfarin comes up, figured out all this. It's natural, it's just the plant is a plant way of killing animals. And so the guys in Holland are really good, Schergers and Vermeer, and what they're trying to do is see, should we be using a mixture of vitamin K and say MK7 from natto or something like that for our treatment. So MK7 gets to all the tissues and can not trash these long-term enzymes and yet interfere. Yeah. But it hasn't been worked out yet. It'll be worked out soon.
What I heard you saying is that for most micronutrients and minerals, eating the right diet seems to get there, and that maybe a multivitamin should be added. With the exception of vitamin D, where you suggested 5,000 units.
Yeah, vitamin D, if you're dark skinned, you should be definitely taking vitamin D. I take a vitamin D, and I get some sun, but not a lot. And my wife works in the garden on Sunday, and but as you get old, you can't make vitamin D very well.
Right.
So I think it's a good idea to have your vitamin D measured. They can measure it, and if you're low, beef it up.
Yeah. My question was beyond vitamin D.
Mm-hmm.
Are there any vitamins that you think are appropriate to take in megadoses, which is often promoted by lots
Yeah.
of
We had a review some years ago pointing out that we found 50 cases in the literature where docs had given high doses of some vitamin and it cured some genetic disease. So for example, there was a case where a rare genetic disease, kids would go blind at a couple of years of age. And a sibling was born, does he have it? Well, somebody tracked down, there was a huge peak of ornithine, which is a precursor to arginine in the blood, and it was some transaminase that was defective. And that's a B6 enzyme. So the doc tried high B6, and now the enzyme worked. So the binding constant is called the Km, and I had a review pointing out all these cases in the literature that when you deform a protein, you can affect binding constant, and sometimes high doses of the vitamin cofactor can now work and make the enzyme work. And I dug out in the literature that the more niacin you give, the higher your NAD levels, the more B6 you give, the higher your pyridoxal phosphate levels. So it's a reason why I never thought high doses made sense, but it's a reason why high doses might work. And it's not only for certain polymorphisms, but as you get older, your membranes are getting stiffer, you're deforming a lot of your membrane protein. So maybe when we're old, we need more of some vitamins and minerals. Anyway, that has to be worked out, but I don't tell people to take high doses of vitamin.
So we're going to call it quits with Dr. Ames. We're going to take a break for 10 minutes before our next speaker, who will talk on thyroidism. Will be presenting. Please be timely. We'll have to leave this room at 10 o'clock. We have to empty out.
Okay, how about one more question over there? He was
So-
seemed anxious.
Oh, you had a question, Steve? You're welcome to leave. Steve has another question, though.
With aging, we also see, for example, changes in hydration, not only during development, you know, the water content in a fetus and a child and all changes dramatically, but with age, we see a drop in water. Do you have any idea whether that's contributing to enzyme activities and binding constants and the effectiveness of various nutrients?
It could be. My wife keeps on nagging me to drink more water, and I think I've been drinking a little more. And so I haven't really researched that literature, but I'll take your word for
But
it.
considering that water is involved in most chemical processes,
Yeah, no,
as
yeah.
your water,
If you get cells more dehydrated, they're in trouble. Yeah.
and the cell walls are going to get harder and more resistant and treated.
Yeah.
All right, 10 minutes.